Hi there, my name's Dr. Onita Basu, and I'm from Carleton University. Thank you for attending my talk today on optimization strategies for biofilters, water quality, and operational adjustments. So first of all, I'm just going to jump into a real quick background and motivation. So in water treatment, we use filtration to help remove fine particles and microbes from solution to help generate a safer water to drink. You can have filters that don't have any microbial action on them, or you can allow beneficial local microbes to develop on the filters, and this would be called biofiltration. In biofiltration, you get filtration and the removal of other dissolved organic substances, which have several benefits, such as reducing disinfection byproducts, taste and odor compounds, bacterial regrowth in the system, and helping minimize membrane fouling if you had a membrane within your system as well. So what's the motivation for this research then? So even though biofiltration has a lot of promise and potential, and many plants are using them, but many plants are not, there's still limited removal of DOC reported at full scale. The range is between 10 to 17%, and I think we should be aiming for something higher than that. The other concern is head loss that develops due to having these beneficial microbes that we've allowed to grow on our biofilters, and this causes an O&M concern for operational staff. So can we make simple adjustments, simple practical adjustments to the system to help improve our biofilter outcomes? So I'm going to present some research from two studies that we've worked on, one at Carleton University in our pilot lab and one at the pilot plant at Ottawa City. The Carleton lab looked at the influence of pH and alkalinity on DOC removal, ammonia, and head loss, while at Ottawa we were focusing in on backwashing methods to see if they could impact head loss development, more importantly, minimize head loss development, as well as the impact on filter ripening and particle release, and then assessing if those had any influences on the DOC and DPB that we observed in the biofilters as well. So here I've just shown a schematic of the Carleton treatment plant that we had. So it's just, we have a water tank. It's from the tap water. It goes through a dechlor column. And then we have carbon and an N&P dosing tank, and from which it goes through a static filter and then onto two identical columns to make sure our results were reproducible. Looks like this in real life, just a real messy lab. You can see the biofilter columns are right here, and our dosing tanks are over here, and an assortment of pumps. For the test itself, we ran from a pH between 6 to 10. We ran at two alkalinity levels, low and high. Those are in milligrams per liter CaCO3. And we used a CNP ratio of 25 to 5 to 1 based on other research I had done. Our backwashing used a pretty standard protocol of a 30% expansion with air scour. The Ottawa City plant actually had 4 columns, and I'm focused in on the biofilter columns here. And importantly, the pilot plant at the city is fed from the full-scale water treatment plant, which comes from the Ottawa River. So instead of using a synthetic water makeup, we were just testing the system with a real water source, which I think has much more practical value to it in the overall study. And then it went through some turbidimeters, as is pretty standard to measure that. We collected the filtered water in some backwash tanks and used them to backwash the columns as well. And this is what those columns look like. You can see them there. And then in terms of the experimental conditions, again, the water was from the full-scale plant. The Ottawa water, as a note, has a CNP ratio of 1,000 to 55 to 1. So it's very nutrient limited. So our DOC, as you'll see later, is somewhat reflective of that. But importantly, our main goal here was to look at the backwashing conditions. So we actually tested 5 of them, water only with a 30% expansion, similar to the Carleton plant, water with an extended terminal subfluidization wash. If you're not familiar with ETSW, That's when you go from your regular 30% expansion and then you use a minimum fluidization velocity. So like think of it as like 1%, it's actually, or less expansion, just to kind of lift those fine particles up and through your filter. We tested the water, the air, water plus air plus ETSW, and then somewhat novelly, the water, the air, and a two-stage ETSW procedure. One note that the two biofilter columns had the same media, but one was more than 50 years old and the other was less than 5 years old. And the media reflected the material that's in the full-scale plant. Okay, so for the first set of results, what I have here is from the Carleton lab, our TOC and ammonia removals with alkalinity and pH adjustments. So overall, we didn't see too much of a trend between or an influence of alkalinity on the results, but we did see an influence from the pH. There's a statistical difference in removal for the TOC at a pH of 10, but none of the other results were statistically different, and I feel like there may have been an experimental error in those results anyways. So the most important key trend to notice is really on the graph is with the ammonia removal. So we have about a 10 to 12.5% removal of ammonia at 6. And this is actually due to the heterotrophic uptake of nitrogen for the cellular requirements of those bacteria at that level. So we didn't really see any nitrification at this low pH in our system from a mass balance that we did. But what we did see in our system is that we were able to develop anecdotally, because this is through measurement and not through bacterial analysis, that we were able to get both nitrification of ammonia, so ammonia removal in the system at a pH of 7.5, which is a good range for that to happen, as well as having maintained our TOC removal, which was really DOC in this case. So this tells us that there may be some opportunities to optimize DOC with ammonia removal in a biofilter system by a very simple pH adjustment. And then just a note that, of course, at the pH of 9 and 10 in particular, at 10, of course, the ammonia is just coming out through volatilization and not through nitrification. And a pH of 9, it kind of, there was, even though it's below the 9.25, there, I think there was a little bit of both happening in the system, but there was still nitrification happening. But that's when we start to see the TOC or the DOC start to drop off. So 7.5 was a better ratio. So moving on to the Ottawa City results, we looked at backwash method and how they influenced head loss. So on the y-axis, I've got head loss, and on the x-axis, I've got time. So our base case is water only. So the water only has this curve to it. And then what we see is that by adding in sort of that extra step, water and ETSW, we have a marginal but noticeable improvement in that head loss development over time. And then here we've got air plus water in the system, the same removal. And again, when we have air and water and one stage of ETSW, it's the same. So any one of those is better than just doing water only. Okay. What's interesting to note is that if we have air, water, and a double stage ETSW, we saw a jump in improvement in that head loss development over time at the end of 72 hours. And so there may be some potential for this to be an improvement in terms of controlling head loss in a system. And overall, the first little group resulted in a 10% difference, and that second one is about an 18% difference. So on this slide, I ended up binning the biofilter for DOC and DPP results. We did an analysis that looked at them by backwash method, and there was no influence of backwash method on any of the results. And so overall, what I wanted to point out was that if you look at the biofilter columns, they had about the same level of DOC removal compared to one of the control columns. And the control column averages 0% removal. And then the biofilter columns are about a 5% removal overall. So not very much, but it's still happening. But if you trickle those results down to the disinfection byproducts, what we can see is that for the THM formation potential and for the HAA5s, when we compared the influent value to the filtered water value, we did see a noticeable drop in both of those values, about 18% for the THMs and 29% for the HAA5s. Okay, so the last slide I wanted to share with you was the Ottawa City turbidity results by backwash method. So here I just wrote regular backwash. That's the water plus air scour, which is the standard procedure that they use at the city. So here what we can see is we've got turbidity on the y-axis and time on the x-axis. And with a standard backwash procedure, we've got our standard filter ripening curve. So we see an initial increase in the turbidity followed by a decrease. And what's interesting is that with the two backwashes with the ETSW and then the two-stage ETSW, we see a decrease in the turbidity curve, which is what we expect. But the two-stage one is better than the single-stage one. And that's really nice to see. But I think we need to do some more testing to confirm those results as well, but it definitely shows some promise. So in conclusion, we saw at the Carleton pilot scale that the pH impacted both DOC and ammonia results, and that the optimal removal was observed at a pH of 7.5 with approximately 65% DOC removal and 45% ammonia removal. And that was also the point with the lowest observed head loss. For the Ottawa City pilot results with the real water, backwashing method did not alter the DOC or DPP removals, which was good to see, which is what we want to see. And a double or stage ETSW had the largest improvement in both head loss development and filter ripening. So that was a quick talk on a lot of exciting stuff. Really want to acknowledge the people with the City of Ottawa that we worked with, several people at Carleton and Humber College, and NSERC for funding this project. Thank you again, and maybe I'll see you at the talking session.